All right, I think we're ready. All right, so there's a little more writing today, so please try your best to keep up. We'll be stopping to talk about the pictures and things, so I'm trying not to move too fast. All right, so we are starting on the intermediate side because if enough oxygen is present, that should be the step that happens right after glycolysis. Okay, so intermediate step. How many pyruvates did we make from glycolysis? Two. Okay, so understand that each of those pyruvates is going to have to go through the intermediate step, and then we'll make two things at the end of the intermediate step, and each one of those is going to have to go through the Krebs cycle. So for what we're talking about today, the numbers that we say of things that get produced, it's basically doubled. Okay. So each pyruvate, which has 3 carbons, is converted to a 2-carbon acetyl group. Now, you don't have to know what the 2-carbon acetyl group, but you need to understand that the way that organisms take in carbon from the environment is as CO2. The way they release carbon into the environment is as CO2. We don't just have little carbon atoms floating around in the atmosphere. Okay? So, that being said, when you are going from a 3-carbon something to a 2-carbon something, the way we get rid of that carbon is as CO2. So keep that in mind for today. That's going to be a running theme, that if I'm losing carbons in my compounds, that's going to have to mean that a CO2 was produced. And so that's number 2. Okay, so to do this, in other words, to go from a 3-carbon something to a 2-carbon something, a molecule of CO2 had to be removed, had to be released. Okay, but remember, we made 2 pyruvates from glycolysis, so each pyruvate is going to have to go through the intermediate step, which means we don't just make 1 CO2, we're actually making 2 CO2s, 1 for each pyruvate or pyruvic acid that entered the intermediate step. Okay, so the acetyl group attaches to coenzyme A to form acetyl-CoA, which is what enters the next major step, the Krebs cycle or the citric acid cycle. So this is a really good picture of what is happening during this step. Notice we form NADH. So tell me what happens to NAD plus during this step. Okay. All right, so if we're giving electrons, if NAD plus is gaining electrons, it had to have gotten them from somewhere. So think back to glycolysis, how I said, look at the previous intermediate or the previous compound. So where do you think NAD plus got its electrons from? Pyruvic acid or pyruvate. So what happened to pyruvate? It got oxidized. How do you know that for sure? Because the step is called pyruvate oxidation. But that's, your answer is probably the more technical answer, which is good. But the step is called pyruvate oxidation, so guess what? Pyruvate gets oxidized. And remember, I do not care if you call it pyruvate or pyruvic acid, does not make a difference to me. All right. And you don't have to copy this part down. This is just something I want you to think about, and maybe you write the answer somewhere on this side of the sheet. For each 6-carbon glucose that you start with, that is a common way for me to start a question, or for the AP exam to start a question about cellular respiration. Because remember, at the beginning of glycolysis, we started with 1 6-carbon glucose. So for each 6-carbon glucose that you start with, how many times does this step have to happen? Twice. Why? Because you made 2 pyruvates during glycolysis, therefore this has to happen twice. So whatever numbers of things you see on here, like it looks like we just made 1 NADH when in fact we made 2. Looks like we just released 1 CO2 when in fact we made 2. And of course we're going to make 2 acetyl-CoAs. Each one of those is going to go through the Krebs cycle. On your posters today, you do kind of show that. You're going to show glycolysis making 2 pyruvates. You're going to show each pyruvate doing the intermediate step. Each acetyl-CoA that you make from this should go into the Krebs cycle, but I'm only having you show 1 Krebs cycle on your poster just for time's sake. What else? Actually, I think in one of your corners, like upper right or upper left, I think I'm having you add in, yes, I'm having you add in an in, out, and where. I don't always do this for the intermediate step, but it seems to help my students. Again, there's no boxes that say it. You just have to write it in some of your free space. So in is what? Okay, 2 pyruvates. You're just writing this in some open space on that side of the page. Something else went in too. NAD plus. Okay. You should have 2 NAD plus that go in. Out, you are making 2 acetyl-CoAs. Two NADH and 2 CO2. And then where? Once you get into oxidative respiration or aerobic respiration, whatever you want to call it, everything happens in the mitochondria. You just got to know what part of the mitochondria. So this happens in the mitochondrial matrix. And I'll remind you where that is. Okay, so mitochondria have that outer membrane. Then they have this inner membrane that is sort of thrown into folds. On those folds that the ETC of respiration is going to happen. But this part that is interior to that inner membrane, that is the matrix. And so that is where both processes that we talk about today, that is where both will happen. Okay, so once you have that down, you can flip your sheet over and we will talk about Krebs cycle or citric acid cycle. So the title of this, the name for this process is going to go in the center of your cycle picture, not at the top of the page like we would normally put it. So in the center of your cycle picture, you're going to write the Krebs cycle, aka the citric acid cycle. Okay, so we are going in numerical order. So find the part that says number 1. And just like we've done with other processes, I kind of narrate what's happening during that phase. And I don't know that you need to be able to draw this from scratch, but you could. It's going to help you if you could practice drawing this at home. All right, so where the blank for number 1 is, this is what you're writing in that blank. Okay, so I know that the coenzyme A just got added to the 2-carbon acetyl group. But the first thing that happens here is that CoA comes off. And so you're just left with that acetyl group. All right, so understand how cycle diagrams work. The 4-carbon oxaloacetic acid, where did you see that before? C4 and CAM plant pathways. Okay, that's oxaloacetate is another word for it. So that 4-carbon oxaloacetic acid is already in the cycle, and your 2-carbon acetyl group is entering. So the 4-carbon acetyl group is going to pick up the 4-carbon oxaloacetic acid is going to pick up the 2-carbon acetyl group, and they are going to combine to form the citric acid. Okay, so that's why one reason why this is called the citric acid cycle is because it is the first stable compound that you make, citric acid. So think about if this were like Heron Circle. This car is already in Heron Circle. You're trying to merge into Heron Circle, and if you were to get in a wreck and combine with that car, that's what you get. Okay, we don't want that to happen, but just that's not meant to mess you up. When you do your cycle diagrams, think back to this. So 2-carbon acetyl group is then added to 4-carbon oxaloacetic acid to make a 6-carbon citric acid, and that's where we get the name of the cycle from. Okay, 3 has a couple different parts to it. Okay, so we know we have a 6-carbon citric acid, but that is going to get changed into a 5-carbon alpha-ketogluteric acid. So when I told you before that I would give you the names of the intermediates, I will. I'll list them for you. Citric acid, malate, malic acid, succinic acid, oxaloacetic acid. I'll list them all kind of in like a little word bank, but you're going to have a diagram to fill in, so you're going to have to know what order they go in and how many carbons they have. Okay? So remember, if we're going to go from a 6-carbon something to a 5-carbon something, the only way we can do that is if CO2 is removed. So that's why if you look on the cart, you see Fruit Loops and Cheerios. The Fruit Loops are going to represent carbons. The Cheerios are going to be oxygen atoms. So whenever you lose a CO2, you're actually going to show that CO2 coming off, and it's going to help give you a visual for this complicated sort of process. Okay, so also during this step, citric acid is oxidized, which means electrons are taken off citric acid and given to NAD plus to form NADH. So let's look at your cycle diagram in the center of your page. Here's kind of what I want you to focus on. Rather than getting overwhelmed by this and saying, oh, I don't know how I'm going to memorize all this, go with the picture. So you see that citric acid has 6 carbons. If you look at alpha-ketogluteric, you can see that it has 5. In order to go from 6 carbons to 5 carbons, you've got to lose a CO2. So that CO2 is going to get released, okay? And in most of these steps, you are making some sort of electron carrier. You're either making NADH or FADH2, except for when you go from oxaloacetic acid to citric acid. So it's kind of a given. And so what I do is I look at that NAD plus becoming NADH, and I say, okay, what's happening to NAD plus? NAD plus is getting what? Reduced, right? Just like you told me for the intermediate step, same thing's happening here. NAD plus is getting reduced. If I'm giving it electrons, it had to come from somewhere. I had to pull them off of the previous compound. So in this case, I'm going to pull them off of the previous compound, which is citric acid. So that's how I know that citric acid gets oxidized. Does that make sense? And so you might say, well, what's the whole point? Why am I making NADH and FADH2? Who cares? Why was it important that I made it during the intermediate step? Because remember what you're doing. Each NADH, those H's carry high energy electrons. Those are not just regular electrons. Why do we care that they're high energy? Because we don't have light in this process to energize the electrons. Our electrons have already got to be energized to start with. So those electrons on NADH are going to have enough energy to make 2.5 ATP per NADH. For each FADH2 that you make down the bottom, they provide enough energy to make 1.5 ATP. So the analogy I used last week was that it'd be like if you're going to Europe and you're collecting currency from different countries, and then when you come back, you can cash it all out for American dollars. What the electron transport chain does, it allows you to cash it all out for ATP. That's why this matters. And that may not click today, but it'll click either tomorrow or Wednesday whenever we do electron transport chain. I forgot to germinate my peas and beans over the weekend, so I'm going to see how they look at the end of the day. If I think we can do the Vernier lab tomorrow, we will. If not, I'm just going to flip-flop what's on your unit plan. You'll still have your quiz tomorrow, but we might flip-flop what we're doing Tuesday and Wednesday if I need to give them another day. So NAD plus gets reduced, and you don't have to write in what I did there because you have the cycle diagram. So we don't want to memorize. If it were me, I'd be drawing this out. I would do 6 little spheres and say, okay, that citric acid becomes a 5-carbon alpha-ketogluteric acid. Give yourself a word bank if you want to, so you don't get overwhelmed with trying to memorize that word. You'll be given those intermediates tomorrow. Okay, if I'm going from 6 carbons to 5 carbons, how's that happen? I release a CO2, and then in most of these steps, all but one, you are making either NADH or FADH2. And then you can talk yourself through what gets oxidized and reduced. If you will take the time to learn it right, not only will you remember it for your test at the end of the week, but you will remember it for your semester exam in December and for the AP exam in May. If you learn it now. If you just try and cram it all in your brain tonight, it is going to be out of there in a couple of days. Definitely out of there before you get to your semester exam. Okay, so if you look at our diagram, now we are going from a 5-carbon alpha-ketogluteric acid to a 4-carbon succinic acid. Since we're going from 5 to 4, another CO2 has to be produced. What's happening to NAD plus in this stage? It's getting reduced. Where am I pulling off the electrons from? What's getting oxidized? Alpha-ketogluteric acid. So you're kind of seeing the pattern. So once you kind of get the pattern, this really isn't that bad. Okay, electrons are taken off of alpha-ketogluteric acid, so it gets oxidized. Those electrons are given to NAD plus to help form NADH, so NAD plus gets reduced. Okay, so that to me is pretty straightforward. You'll notice there is a step C under number 4 because it shows you even in this picture that ATP is made, but it's not going to be as straightforward as what it looks like there. I added a little thing, a little picture to the bottom. So let's think back to Friday. The last thing that we talked about Friday, I believe, was the difference between substrate level phosphorylation and oxidative phosphorylation. Substrate level, you are pulling a phosphate group off of another compound and adding that phosphate group to ADP to make ATP. If you're doing oxidative phosphorylation, it is probably happening during an electron transport chain. You are making bunches and bunches of ATP, and you are using just free inorganic phosphates that are not attached to a compound. Look at the little picture that I added in. Okay? It has GTP and GDP on it. What does it look like is happening there? Yes, it is stealing a phosphate group from GTP and adding it to where? ADP to make ATP. So based on that description, are we doing substrate level or oxidative? Substrate level. So what letter C is showing you? Because remember, I told you in notes Friday that substrate level phosphorylation happens during glycolysis and Krebs. And I showed you a very small example of the step where it happens in glycolysis. This is crystal clear. This is substrate level phosphorylation during Krebs. Oh. And you don't have to write the question down here. I would just maybe star and tell yourself that this is substrate level phosphorylation. So during this step, a phosphate group is taken off of guanosine triphosphate, which is GTP, and that phosphate group is added to ADP to form ATP. So this is substrate level phosphorylation. It happens during glycolysis and it happens during Krebs. Okay, so let's look at the next step. We are converting a 4-carbon succinic acid to a 4-carbon malic acid. You've heard of malic acid or malate before. Do I need to produce CO2 this time? No, because I'm not losing a carbon. Okay, and I'm not going to do it the next time either because oxaloacetic acid is 4 carbons. So you've made all the CO2 you're going to make this cycle. All right, we are going to make a different electron carrier. It is FADH2. Even though there's 2 hydrogens instead of 1, those hydrogens actually store less energy. So the electrons that we pull off of FADH2 are going to start lower in the electron transport chain than the electrons we pull off of NADH. You'll see that more either tomorrow or Wednesday, whatever day we do the ETC. Okay, so 2 hydrogens are removed from succinic acid, so succinic acid gets oxidized. This forms a 4-carbon malic acid. The electrons are added to FAD to form FADH2, which is another electron carrier. It's just that its electrons don't store as much energy as the ones in NADH do. All right, so if we look at our next step, we've got a 4-carbon malic acid becoming a 4-carbon oxaloacetic acid. So do I need to make any CO2?
No.
Nope. You're just going to make a little bit more NADH. So electrons are removed from the 4-carbon malic acid. So when we say that NAD plus gets reduced, remember, always look at the previous compound because that's what's getting oxidized in that step. So we're pulling electrons off of malic acid and we're adding them to NAD plus to form NADH. And this is going to form the 4-carbon oxaloacetic acid that we started with. Okay, so I think we're just, once everybody's got that down, we just need to do our in, outs, and wheres. So try to be coming up with those because you do need to know numbers for this. Got to know how many of each were produced. How many times is this cycle going to happen? Twice, right? Because you made 2 acetyl-CoAs from the intermediate step because you made 2 pyruvates from glycolysis. So any numbers, or if you count up the number of things that get produced in your picture, we would have to double those because this is just for 1 turn of the citric acid cycle, not 2. Okay. All right, so our ends. Our major N is acetyl-CoA, but our minor Ns are also important. That would be GTP, FAD, NAD+, and ADP. All right. First, remember that this cycle happens twice. How many CO2 should be produced total? 4, right? So we're going to write 4 CO2. All right, how many ATP? Just 2. How many NADH? 6. 3 per turn. And then FADH2? 2. And this is also taking place in the mitochondrial matrix, just like the intermediate step did. All right, while you finish that, I've just got to go move a couple tables in C312. I'll give you some brief instructions. So stay at your desk a second.
Cut 4 more pieces of poster paper because that's what we need for your class. And then I'll give you some brief directions. You'll have really about half the period to finish your book. Okay, so here's the plan. Let me shut off my recording first. There are 13 big tables in there, so you'll have enough room, I think, to work pretty easily. That means we are all in pairs, so we do not need any groups of 3 today. It's just for today. You can just work with somebody. It's not gonna